Welcome to CROssBAR version 2. CROssBAR version 2 is a unified biomedical knowledge graph designed to integrate heterogeneous biological data into a single structured system. In modern life sciences research, relevant information is scattered across many databases, making integration and analysis challenging. So we address this problem by bringing these diverse datasets together within a coherent Neo4j-based graph architecture. Here is the overview of the CROssBAR version 2 knowledge graph. It integrates 34 unique databases, covering 14 node types, including proteins, genes, pathways, diseases, drugs, and phenotypes, and 33 relationship types. The graph currently contains over 2.7 million nodes and more than 12.6 million relationships. CROssBAR version 2 can be accessed in multiple ways. You can interact with it through natural language using CROssBAR-LLM. You can access it programmatically via our GraphQL API. Or you can manually explore the graph using the Neo4j browser. Together, these access layers make CROssBAR version 2 flexible, transparent, and adaptable to different research workflows. Now, let's put this knowledge graph into action with CROssBAR-LLM. Instead of writing complex database queries, you simply ask your biological question in plain English. The selected language model then generates the corresponding cipher query, executes it on the knowledge graph, and returns the results as a clear natural language explanation. To get started, you can select one of the example queries displayed in the right panel, or enter your own biological question directly into the chat. When writing your question, here is an important tip. If you are referring to a biological entity such as a gene, protein, or disease, type the @ symbol and begin entering the entity name. This activates the autocomplete feature, which searches the knowledge graph and allows you to select the correct entity along with its type. To configure the model used for your query, open the Model Settings menu in the right panel. You may use one of the free models provided without an API key. Or select another model and supply your own API key. When submitting your question, you have two options. You can choose Generate Only. This creates the cipher query but does not run it. You can view the generated query in the right panel, copy it into Neo4j browser, modify it if needed, and run it manually. Or you can choose Generate and Run. In this case, the system creates the query, executes it, and returns a natural language response. For each response, you can view the details in the right panel. There, you will find the generated cipher query, the retrieved biological entities, links to their source databases, and the structured JSON output. CROssBAR-LLM also supports iterative exploration. After receiving an answer, you can continue by asking a follow-up question. You may type your own follow-up or select one of the 3 suggested questions generated by the system. Your conversation history remains visible in the chat, and you can revisit the details of any response at any time. In addition to direct graph-based querying, CROssBAR-LLM offers optional vector-based semantic similarity search. By enabling the vector-based similarity search option, the system leverages pre-computed embeddings to identify biologically related entities based on semantic similarity. In this mode, you can select the embedding type corresponding to the biological entity of interest and upload your own vector representation. Or use one of the provided examples with their preloaded embeddings to test the system. The results can also be explored further through follow-up questions. Thank you for using CROssBAR-LLM. We're excited to support your discoveries.